An organ is a structure composed of two or more tissues which performs a specific function. When observing tissue slides, it is important to recognize that very often one is observing organs which contain multiple tissues. And so a student who takes a specific slide and says that I can't find the epithelium might be looking at the wrong part of the slide and might be focused on, say, muscle tissue instead. So, for example, in this slide of the trachea and the esophagus, the trachea is composed of multiple tissue types since it is an organ. It is lined by pseudostratified columnar epithelia. It possesses C-shaped rings of hyaline cartilage, which is a connective tissue. In the posterior of the trachea, a smooth muscle band known as the trachealis controls the diameter of the trachea. And then there is also areolar connective tissue and adipose in the vicinity as well. The esophagus is lined by stratified squamous epithelia. There is connective tissue in its submucosal layer, in addition to the serosa layer, the thin layer of collagen fibers which wraps the esophagus as part of a serous membrane. In addition, there is a muscularis layer which contains skeletal and smooth muscle, depending on which part of the esophagus is sectioned. Around these structures, adipose serves as a packing material. There are multiple blood vessels in the area, and blood vessel linings contain both epithelia in the endothelial lining, smooth muscle, and connective tissue. And nerves will innervate the trachea, the esophagus, and the blood vessels. And so there are multiple tissues represented, and a student should be familiar with a structure prior to examining it under the microscope to make sure that they are actually observing the tissue they mean to.